Welcome to the Bio and Beyond podcast. I'm your host, Sawik Nath, and today we're diving into the world of biomedical engineering. Please join me in welcoming our distinguished guest, Dr. Vlaminck. Dr. Vlaminck is an associate professor of biomedical engineering at Cornell University. His research is focused on the development of precision medicine technologies to monitor and study infectious and immune-related diseases. His research was recognized with the NIH New Innovator Award, the Noyce Foundation Assistant Professorship in the Life Sciences, and Rainin Foundation Synergy Award. He received Teaching Excellence Awards from the College of Engineering in 2017 and 2022. He is a co-founder of Canvas Biosciences. He has a tremendous amount of contribution to the field of BME, which includes research and teaching. Today, he is here to speak about his work and to inspire others to take interest in BME. Without further delay, let's dive right in. Why don't you tell us a little bit about yourself?
Well, thank you. First of all, thanks for having me on your podcast. Happy to chat with you. I grew up in Belgium, moved to the US about 12 years ago or so. And I got into BME via not really a straight path. When I was young, I was interested in, so many students interested in mathematics and the sciences. And I followed an engineering degree, civil engineering actually. And I really got interested in research because of the opportunity to create something new, to invent something. The process of invention was really something that spoke to my imagination. And during my PhD research, I worked a bit on sensor technologies, small devices that could interact with human cells and molecules in order to detect them. And that made me think about biology, but obviously I didn't know much about it. So I decided if I wanted to go in that field, I needed to learn about biology. So I did actually two postdocs. I first went to the Netherlands from Belgium to learn about biophysics, and I worked with Kees Dekker, developing novel tools to learn about DNA protein interactions. And then later on, I moved to California to do a postdoc with Steve Quake at Stanford, where I got interested in genomics. And then after that, I was very lucky to be able to launch my own lab at Cornell University now about 9 years ago. And so my lab is very interested, as you mentioned, in it's a biomedical engineering lab. We're interested in developing tools to learn about infectious disease and microbiome-related disease, immune-related disease. And it's been really interesting and rewarding.
That's really interesting. So you said that you came from where did you say?
Belgium.
Belgium. So
Yes.
how would you say that it's different learning there as compared to here?
I don't think it's so different. I think what is really great about the US environment here at Cornell and at other places is that undergraduate students are involved in actual research much earlier than back in Europe. So that's certainly something that I think was missing in Europe. And in fact, actually, it was only when I was in my last year of my undergraduate studies that I got an opportunity to work in a research lab. And I have to say that really sparked my interest in pursuing research in the sciences in general. And it also just made me a better student. So I think the fact that in the US, students are involved in research and science so early on is really wonderful.
That's really interesting, and it's a good thing that students are involved with the sciences from early on because then they get more experience, right?
Absolutely, yeah. And you learn to think about science in a completely different way, which I really enjoy about being a PI and being a researcher in general, is that, the way you think about research when you try to develop something new, discover something new, invent something new, your mindset is very different from when you're just trying to understand what is already known. And so I think that switch in thinking is incredibly enriching.
Okay, well, let's move on. Would you like to speak on some of your recent research?
Yeah, absolutely. As I mentioned, we're interested in developing measurements for biology and medicine. Let's say we are interested in developing new approaches to learn about human biology and potentially to develop new diagnostic approaches. And there's a couple of things that we've been doing in the lab lately that I'm very excited about. The first big topic in the lab is focused on profiling, measuring molecules of DNA, but also RNA that circulate freely in blood. These molecules are released in the bloodstream by dying cells, and you can recover them from the blood and then analyze them using advanced DNA sequencing approaches, for example, to learn a great deal about where these molecules are coming from. What we found is that is an incredibly powerful approach to learn about immune disease, but also infectious disease. And so we've been pursuing that. We've been thinking about new applications. We've been thinking about new data analysis approaches, AI and machine learning and things like that, which I know you're interested in. And then we're also interested always in developing better molecular tools to retrieve as much information as we can. And the other major topic in the lab now is to develop imaging, but also sequencing-based approaches to learn how tissues are organized. So what biomolecules are present in tissues and how they're spatially organized. Because we think that those kinds of approaches can teach us about how tissues work and how tissues develop and how tissues change in response to disease, for example, infection. And that's also been incredibly powerful and fun.
Could you explain? So the first one, you said that you found molecules in the blood, and then you took those and found out like the immunohistory and like things like that. Could you explain? Could you give a short explanation on how you do that? I think that's really interesting.
Yeah, so we didn't really discover that. That's actually something, the fact that that nucleic acids, DNA and RNA, are present in blood plasma is something that had actually been observed already in the 40s by two French scientists, even before the structure of DNA was known. So this knowledge goes way back. But the implications of that, in particular for measurements of human biology, are only now sort of becoming clear. And so what this is focused on is short pieces of DNA and RNA that are present in blood outside of cells. So they're called cell-free DNA and cell-free RNA, not contained within cells. But they're coming from cells, of course, and their origin is really due to cell death. So whenever cells die in your body, cells in the blood system, but also cells in your tissues, whenever they die, and cell turnover is a common process in biology, not only disease-related. Your lungs are about 7 weeks old, for example. Your tissues are constantly renewing, most of your tissues. So whenever that happens, whatever is inside of these cells doesn't vanish magically. It can end up in the bloodstream. And so these molecules can stay there for some time, not very long, are rapidly cleared, but you can retrieve them from the blood and then analyze them using the incredibly powerful DNA sequencing technology that now exists, right? You might have heard, about the Human Genome Project and all the advances in DNA sequencing. Those processes have now become incredibly robust, but also incredibly cheap. You can sequence 1 billion molecules for $1,000 or so. So you can capture an incredibly rich picture of what's going on in a person's body by analyzing these molecules of DNA and RNA that are sort of the detritus of all the cell death processes, development of processes, disease processes that might be happening in that person. So that information is in theory there. And then we are interested in developing tools to actually obtain the information. So to extract those molecules of DNA and RNA, to analyze them in a way that is as unbiased as possible. And then, of course, to also analyze them. It's not easy because the data sets tend to be very large. So that's been incredibly fun.
Yeah, that sounds really interesting how you can just do that. And once again, talking about the Human Genome Project, you said that now it's incredibly easy to access that information.
Yeah.
Compared to long ago, which is really interesting about how much science has advanced from that time.
Yeah, absolutely. It's been incredible. Bioengineering and biomedical engineering are advancing incredibly quickly. There's never been a better time for students that are listening to this podcast. There's never been a better time to enter this field because the possibilities are just mind-blowing. I mentioned the Human Genome Project. that the first draft, an early draft of the human genome was published in the early 2000s, 2001, I believe. And to complete that draft had taken many years by many investigators. And it cost about $100 million or so. And nowadays, it's possible to sequence a human genome for a few $100s, and you can do it incredibly quickly. And so that's great for people that study the genome, geneticists, but it's also great for people like us who like to think of new ways to measure biology and try to think of new ways how we can use that incredible resource, these incredibly powerful devices that can read out the sequence of DNA molecules with incredible accuracy. How can you use that to learn about what's happening in a person that is dealing with infection, or to learn about what's happening in an organ that is infected with a virus, for example?
Yeah. Well, anyways, moving on to my next question. Can you share some other important works that are happening in the field of BME at Cornell?
Yeah, I mean, there's a lot going on. The department is extremely lively. I have colleagues that are working on the microbiome. I have colleagues that are working on developing new imaging approaches for biology and biomedicine, to look at phenomena that are happening in real time in the brain of mice, for example. I have colleagues who are interested in engineering tissues or building organs from the ground up, which is an incredibly inspiring field of study. So there's lots going on.
Yeah, that's really interesting how wide the field of BME is.
Mhm.
Like, what you could be doing is completely different to what someone else could be doing in BME.
Mhm.
It's like completely different.
Yeah, exactly. But that means there's also a lot of opportunity for collaboration, right? So I have colleagues who are interested in how muscles repair after injury, for example. And some of the approaches that my lab has been developing are in principle incredibly powerful to learn about those processes. So there's natural ways to collaborate across these different disciplines, and that's also really exciting.
Yeah. Uh, so are there any innovations like machine learning or generative AI that are changing biomedical engineering?
Yeah, that's something we're really interested in at the moment. Of course, there's been well publicized breakthroughs, like, for example, AlphaFold, that has been used to learn about protein structures, for example, and how proteins interact with one another more recently. It's also interesting just to think about how large language models in general can impact the way we do our science, right? Or the way we analyze our data. And so we're very interested in those approaches as well. So I think it's true to say that biology and biomedicine, biomedical engineering are sort of become computer sciences, and that's due to, first of all, the availability of huge amounts of data. Right, we can learn an incredible amount about a biological specimen now that was not possible before. You can extract incredibly high-dimensional data from a sample. And then at the same time, there's all these advances in computer science that may impact how we analyze the data, how we interpret things in biomedicine. So again, I just want to say that it's an incredibly interesting time to be doing work in this field.
Do you know anyone who currently is using uh, machine learning in the field of BME?
Oh yeah, there's many labs, and in fact, I think I would say that most BME labs are interested in incorporating some of those elements into their research. Engineers are just, of course, always looking for new ways to make progress and to incorporate new ideas into your research. One of the reasons I love to do research is that it's an incredible opportunity to learn by doing. So absolutely, we are doing that. I mentioned the projects where we're looking at RNA and DNA that floats in blood. Again, that gives us incredibly deep profiles of these molecules in patients, and we do this across large patient cohorts. And of course, we've started to use machine learning approaches to make sense of that data, right? So there's these advances in machine learning, and maybe AI, that's just something that we're now thinking of. Will enable just incredibly powerful ways to find patterns in those data that can then be turned into basic knowledge of particular diseases, but could also be used to develop diagnostic tests. And so we're interested in both the basic science questions as well as the potential applications that may come out of this.
Mm-hmm. Yeah, that's really interesting because let's talk about before all of this machine learning came to be. People were finding advances in a lot of different ways, but now that we do have machine learning, the advances are coming a lot faster and people are learning a lot more.
Yeah, that's probably true. I will say that the quality of the data is also a determining factor. So I think we not only do we need advances in the way we analyze the data,
Mm-hmm.
and what kind of data we analyze, but we also need new advances in how the data is generated, how the data is produced, so that you can then consume it using machine learning approaches, and then implement it somewhere, hopefully, so that it impacts human health.
Yeah, that's really interesting. I find all of this extremely interesting. So, where do you think that where do you think that the field of BME is evolving to in the next 5 to 10 years? Where do you think it'll be?
Yeah, I mean, as we've mentioned, the field of BME is hugely diverse, right? There's a lot going on, so it's hard to give one answer. But I will say that the major trend that I see that I'm excited about is as I mentioned, that this field is also becoming a computer science. That these data analysis concepts are seeping into the field now. That people are coming up with new ways to analyze the data, and also the fact that there's these incredibly powerful ways to extract very deep information about biology that are just coming to the forefront now. To me, that is the combination of the ways data are generated and then analyzed is incredibly powerful. My lab has not been active in the field of synthetic biology or manipulating biology, but of course, there's also been incredible advances in CRISPR technologies and related technologies to manipulate biology. So not only do we have incredibly powerful and increasingly powerful tools to extract information from biological systems, we have algorithms to analyze that, but we also have incredible powerful ways to manipulate biology. And so that combination is just incredibly rich.
Mm-hmm. Yeah, that is true. Now that we have so much, we can probably advance so much further than we did in the past 10 years, or in the past 10 years, in the next 10 years. Do you think, how far do you think that we're gonna go? Do you think that we can, what do you think we will be able to do that we haven't been able to do in the past?
Well, I mean, I think every area of science, certainly every lively area of science, continuously accelerates. So as you point out, the pace of innovation, the expectation is that the pace of innovation in the next 10 years will be higher than the pace of innovation in the previous 10 years. And that should make a lot possible. I think we'll learn a lot more about human biology, human disease. At the same time, if you're working in this field, biology is incredibly complex. It's humbling. And so I do think that as we progress, the picture that will appear is that the complexity of biology is only just now presenting itself. So I think we need to stay humble. In the next 10 years, we won't solve all problems.
Mm-hmm. So, what advice would you have for someone who is thinking about going into the field of BME?
Mm-hmm. Well, just I would say follow your instincts, follow the science. I think if you enter this field, it cannot be a bad choice. There's so many possibilities in research, but also in industry and in just the medical industry. So I think it's if you're interested in biology, you're interested in engineering, it's a great opportunity to do something that can impact human health, which ultimately is incredibly rewarding. So I would just say go for it. Work hard and again follow the science. Ask yourself, where's the most interesting science? Where's the most interesting research happening? Where can you learn the most? And then take it day by day, and good things will happen.
Mm-hmm. What would you say was the biggest struggle you had in your time as a BME expert, and how did you overcome that?
Um, I think that the science is very difficult, right? Uh, because of the complexity of biology. First of all, it's hard to predict which projects will bear fruit quickly and which ones will be difficult. Sometimes I'm always optimistic, certainly when it comes to technology.
Mm-hmm.
I should be more optimistic in general, but certainly when it comes to engineering and technology, I always my intuition is always that things uh, will work out. But sometimes it takes more time. Sometimes, sometimes you have an idea, but it really takes several years for it to come to fruition. And the way to solve that or deal with that is in a way is it's good to deceive yourself, to be able to convince yourself that the solution is just around the corner, um, and and to stay optimistic and just work day by day. And I've always been sort of I've always had the sense that things will work out soon, and then sometimes this happens only 5 years later, uh, but then it's still something that you can look back on with a lot of pride. So yeah, just the struggle is that science and research can be very difficult, and it doesn't always work out. Sometimes sometimes projects. Now I'm in a position where I can do many things at the same time, and sometimes projects just click very quickly and fall into place. But it doesn't always work out that way.
Okay. Well, last question. What would you say are the three biggest values that you have as a biomedical engineer?
Um, I believe in innovation and invention. It's really what drives me, I think. The things that I get excited about are really coming up with new concepts to make something new happen. I find that incredibly inspiring, even if they're smaller advances. Yeah, that always gets me excited. Um, I work my lab. Um, is mostly consists of postdocs and students who do the research. Um, and I believe in open communication, frequent communication, free exchange of ideas. So I try to promote that. And then lastly, I think it's incredibly important for a lab, certainly biomedical engineering, but probably other areas of science as well, is to have diversity of opinion and have diversity of personalities. And so our lab has been very fortunate at Cornell. We have great students in computer science, computational biology, physics, biophysics, biomedical engineering. And so uh, my lab has students from all of these different areas of research. And that makes for a really interesting place because some students have a deep understanding of molecular biology, other students have a deeper understanding of statistics or computer programming. And I really think that sort of building this kind of interdisciplinary group has been very rewarding. But I also think it makes for better science. And it's not only in terms of expertise, diversity in expertise, but also diversity in backgrounds. We have students from all over the world, from different walks of life, and they all bring their own perspective. And so that's an important value as well, I think.
Okay. Well, before we end, do you have anything else you'd like to add?
No, I'd just like to thank you again for doing this. It was great to chat with you, and thanks to the audience that were listening. I don't know who your audience are, but
Mhm.
um, yeah, thank you.
Okay. Well, as we wrap up this fascinating episode, I'd like to thank Dr. Vlaminck again for sharing his incredible knowledge and expertise in biomedical engineering. His dedication to the field is truly inspiring. We'll continue these captivating conversations at the intersection of science and innovation. Until next time, this is Satwik Nath, signing off from Bio and Beyond podcast. Remember, the exploration of science never ends. There's always more to discover.